warm welcome to everyone. I'm Stalin, and I'm currently doing BPharm at KMCH College of Pharmacy, and I'm here to present a manuscript. The topic of tier proteomics reveals the molecular basis of the efficacy of human recombinant nerve growth factor in the treatment of neurotrophic keratopathy. About the author, Damiana Pieragostino is a corresponding author who is working in the Department of Medical, Oral, and Biotechnological Sciences. Let's dive into the content. The aim of this study is to explore the molecular mechanism underlying the efficacy of recombinant nerve growth factor in the treatment of neurotrophic keratopathy by using the tier proteomics. It helps us to identify the protein which is expressed in the treatment and also helps us to identify the potential biomarkers for the monitoring the therapeutic response. Tear proteomics. Tear proteomics is the study of proteins present in the tear fluid by using the proteomic technologies and also help us to understand the protein composition which will change according to the disease and treatment. Neurotrophic keratopathy is a rare degenerative corneal disease and it is coming under the orphan category of diseases and it is characterized by the reduced corneal sensitivity and poor epithelial healing due to impaired corneal innervation. This neurotrophic keratopathy is categorized into three stages. Stage 1, the starting stage, mild epithelial changes will occur as a result of punctate keratitis. In stage 2, the persistent epithelial defect, and the stage 3 is a severe condition in which the corneal ulceration, stromal melting, and perforation takes place. For addressing this issue, human recombinant nerve growth factor which is used for the neurodegenerative diseases like the neurotrophic keratopathy by promoting the neuronal growth, survival, and the regeneration. It is approved in the treatment of. It is approved in the Europe for the treatment of neurotrophic keratopathy. Its mechanism is based on the promoting the neuronal growth and the survival, and also it promote the epithelial healing by reducing the apoptosis and enhance the epithelial cell proliferation and migration. So I divided the methods into 5 steps. The first one is the patient enrollment and clinical evaluation. Total 50 patients are selected for the treatment, among them 7 males and 8 females. So they were administered Senagermin is an eye drop which contains human recombinant nerve growth factor. They were administered a single drop every 2 hours, 6 drops per day until the end of the treatment. Only 8 patients were completed the therapy and they were evaluated by the slit lamp and the Schumacher's and in vivo confocal microscope. The sample which is collected at the university clinic is stored under minus 80 degrees Celsius followed by the FASP protocol and the proteomic analysis is performed by using the liquid chromatography, the tandem mass spectroscopy, and high resolution time of flight mass spectrometer. And the data were processed by using the MaxQuant software with the help of Unicode database. And the Perseus is the bioinformatics tool, which is used for the interpretation and the protein quantification. And the log2 transformation is used for the data handling of the upregulated and downregulated proteins. And the volcano plot, the t-tests were used for the statistical analysis to identify the significant. And the data which is stored in the proteome exchange consortium, the data identified is given below. Coming to the results, so in proteomic analysis, in figure 1a is a Venn diagram which clearly indicates the common and unique protein present in the three different clinical conditions. T0, T4, T8. T0 is the baseline level, T4 is the at the 4th week of the treatment, and T8 at the 8th week of the treatment, that is the end of the treatment. 3 unique proteins were identified in the base level, and 2 unique proteins were identified in the T4 level, and 6 unique proteins were identified in the T8 level. So in figure 1b, it's a Volcano plot. It's used to compare the ratio of protein expression between the T4 and T0 level. The blue dot, which represents the protein that are not differentially expressed, and the red dot which represents the protein which are differentially expressed. The red dot which present on the right side is indicates the upregulated protein, and the red dot which presents the left side of the plot which indicates the downregulated protein. In figure 1C, the protein expression ratio is compared between the T0 and the T8 level, and in figure 1D, the protein expression ratio is compared between the T4 and T8 level. So these are all the unique proteins which have been identified in the three different clinical conditions at baseline and T4 and T8. The plus sign indicates the presence of the proteins at three different conditions. Extracellular vesicles. These extracellular vesicles play a pivotal role in the neuronal regeneration and the immune response and also the tissue repair process. There are three extracellular vesicles which have been identified: CD171 plus, which presence indicates the nerve regeneration activity, and the CD126 plus presence indicates the inflammatory signaling, and the CD45 plus indicates the ongoing immune response and the tissue repair process. In the figure 2A, which is a bill plot, which is usually to which is used for the relative distribution of CD171 plus among the three different clinical conditions T0, T4, and T8. An asterisk symbol is used for the significant reduction. And inside the each bill plot, there is a scatter plot which indicates the individual measurement of each patient who have different composition of the CD171 plus across the treatment. In figure 2B is a rank correlation graph which correlates the rank between the CD171 plus and the CD126 plus. And the PSN value coefficient value is 0.71. It indicates strong correlation between the CD176 plus and the CD171 plus. In figure 2C, this is the dot plot, which is used to identify the people who have double positive CD171 and the CD126 plus 126. And it helps to distinguish and quantify the subset of CD171 and the CD126. In figure 2B is similar to the figure 2B. It is also a rank correlation between the CD45 plus and the double positive CD171 plus and the CD126 plus. The coefficient value of 0.56 clearly indicates the moderate strong correlation between the two sets, and it clearly indicates the extracellular vesicles play an important role in the nerve regeneration and the immune response. So this figure represents the this figure has have been taken before and after the treatment of human recombinant nerve growth factor. This clearly shows the person who undergone the treatment who have the increase in the nerve fiber length, density, and the number of branches during the treatment. So it clearly showing that recombinant human nerve growth factor effectively promote corneal nerve regeneration. Coming to the discussion, the efficacy of the recombinant nerve growth factor, this study showed that the treatment significantly alters the tear protein expression in the patients. It reflects the mechanism which is behind the clinical improvements. And also, it indicates a dual role in the corneal healing process through two pathways, BDNF and IL6R, and it is validated by using the Western plotting and the flow cytometry. And the validation of the increased MMB9 expression and the analysis of the extracellular vesicles supported the proteomic finding and suggesting the neuronal derived extracellular vesicles CD171 plus as a potential biomarker for the treatment response. So coming to the conclusion, this study elucidates the molecular mechanism which underlying the efficacy of human recombinant nerve growth factor in treating neurotrophic keratopathy by highlighting the significant changes in the tear protein expression, the observed modulation of the inflammatory and neuroregenerative pathways validated through the advanced proteomic techniques underscores the recombinant nerve growth factor play a dual role in the corneal healing. These findings pave the pathway for the improved patient management and personalized treatment strategies. Thank you.